can also evaluate for the specificity in terms of, because nowadays there are tools which are being developed even in the software assessments. We spoke about alpha beta assessments. Nowadays people also see area overlaps. So although this being a qualitative assessment, so they will see how much area is overlapping, and the percentage of area overlapping between the RLD and our molecule can also be termed as a specificity between the structural assessment. So yes, it is tricky, but it's not impossible. You can also do spiking like we do for other physicochemical methodology, where you have a high alpha, high beta, high random coil structure and do a spiking and show that at what levels can your CD detect these. But of course, all of this is possible only when, along with the optimization of the methods, when we are using these kinds of samples during the development itself. Now the USP chapter also talks about the life cycle management, where the QBD approach for the analytical method development also has come into the picture. So all of these may help us in defining some sensitivity in the CD as well. I hope that answered part of the question.
Thanks for that. I have another question. Whenever we do the FAR CD specifically, we dilute the sample to specific, preferably to 0.1 milligram per ml. If your drug product concentration, let us for sake of understanding, we can consider 40 or maybe 100 milligram. So when you are diluting this to 0.1 milligram, how do we ensure that the structural changes are not happening, and what we are reporting is the true representation of your sample?
Yes, as I again projected, there are some limitations of the concentration till which you can go. So within the ambit of that limitation, you can do a linearity set to prove that there is a saturation level. Like we saw from 0.1 mg per ml till 0.25 mg per 0.2 mg per ml. We used different concentrations to show that the sensitivity or the linearity was maintained. So you can use that. But if you are trying to evaluate 10 mg per ml neat versus 0.1 mg per ml neat, as of today, the CD does not have the capability to do that. We face this challenge in size exclusion chromatographies as well for any aggregation pattern. You do a neat injection or you do diluted injection, but then there are some tools to study. Like we are using a UPLC, you can do both. For the SEC, like you can do a neat injection, you can do a diluted injection with various diluents and see the impact. But unfortunately in CD, there is a limitation of concentrations. Now, if you go to the tertiary structure determination using CV, that requires a higher concentration. That cannot be done at 0.1 and 0.2 mg per ml. That has to be typically done more than 2-3 mg per ml. So both of them has their own limitations.
I agree with you, because the proteins are mostly it is observed that the proteins are stable at higher concentrations. When you are diluting, that too you are changing the buffer, because your formulation excipients may not be compatible with the CD. So you have very limited choices like PB and all those things. So you don't know when you are diluting how it is happening. So I understand the way you have demonstrated the linearity. Basically, that is the one of the aspect where we can optimize the concentration. But it is difficult to ensure that the whatever we are reporting is the true representation of the sample. And I understand this is a limitation. And specifically when we are demonstrating the biosimilarity, it gets nullified, because if it is happening at all, it would happen for comparator also. Yes. So yeah, thank you. Thanks for that.
NMR for that, and that is why these techniques are orthogonal. Actually, NMR holds an advantage over there, because higher the concentration, you get better sensitivity. So, thank you.
Hi, thank you for the talk, and myself Smita from Merck Life Science. So my question is with respect to the high concentrated MAP formulation. So viscosity is a major challenge when it's but I could not see the test for the viscosity into the analytical methods what you have enlisted.
Well, most of the if you are talking specifically about the monoclonal antibody high concentration formulations, most of the antibodies available as of now does not have that kind of a viscosity issue. They are all easily miscible in water and they are aqueous in nature. So not as such, but yes, nevertheless, as I said, these are not only limited to tests. There are so many other things that you can do. We didn't speak about the density assessment of we didn't speak about extinction coefficient determination. So all of this can be done, including the viscosity, just to understand if it is matching, or how batch to batch also,
correct,
or scale to scale also, or volume to volume also it differs. So yes, definitely, it's not limited to the slides that we have presented in any ways.
Okay, fine. And another question is regarding the nano DSF. So whether you think that this technology is also promising for high concentrated maps?
Well, yes, short answer to that question is yes. Any new technology is always promising. We have not studied it extensively, so I cannot comment on the particulars of nano DSF, but the claims is that it can do almost everything that a CD can do. and much more.
Yeah.
So specifically in terms of melting point determination, so it also can do what a DSC can do in short. So it is a combination of two techniques if you see that way. But technically we have not evaluated, so I cannot comment on that. But yes, it just looked like an interesting tool.
Okay. Thanks, Pradeep.
Hello, I'm Supriya from Genex Genomics. I wanted to understand, like, when we are talking about orthogonal approach, so during the product life cycle, how do we understand that which approach would be better at what time point or during the cycle?
Okay, so good question. So this is future need of FDA. So whenever initially we started with overlapping, only spectral overlapping of submission. Then gradually they're asking, going on the numbers. So how similar it is. Nowadays they're asking, furthermore multiple technology. As he's rightly stating, Dr. Pradeep, all the technology is very, very important from different perspective because it gives its own, overall picture of your product quality. So that's the ask from the FDA. So what they ask is overall, you have to have as many as orthogonal techniques which would prove your product is very much similar to your innovator. That's the whole purpose. So in any one technique, if you are differentiated, then that will going to do. That's what the innovator do. So if you look at, I don't want to take a name of a couple of products where they have a team to have a differential analytical techniques to show that any generics would show little bit differential from my innovators. They show that's called citizen petition. So if you take a glatiramer acetate, it's like a saga, after more than 10 years of a third and fourth citizen petition, FDA themselves came out with a guidance document stating that let's do this much. So this is how. So they just protect. It's money game. Okay. And the other important point, I just wanted to add on in here is now FDA is also asking kind of stress studies. I mean, initially we were thinking that okay, let's do only initial level, a comparability study at initial level with all 30 techniques. Not like that. Now they're asking, they call it as a stress, but there is a multiple condition over there. So what it implies is exactly what is shown in the slide. If there is a difference in any of the chromatographic parameters, any of the structural parameters, that you will going to catch up. So then you have to prove whether that would eventually lead to safety or immunogenicity kind of a thing.
Just to add to that, give a closure to your question, the earlier concept was orthogonality at the end product. But the current requirement is orthogonality right from the start of process development. So at every angle, if you use orthogonality, you are going to control these residual differences at a very early stage of product development itself. So maybe at the end of it, even though the absolute values of different methods will be different, but the relative values between the RLD will be consistent. So that is the new ask, and that is a mandate now.
Yeah, before we take on the next question, I would like to introduce two more members here. Dr. Ravishankara, and he is currently working at Sunpharma Industries, and he is leading the team for analytical division. And then we also have Dr. Vadiraja Bhat. I've known Vadi for a few years. He is working at Agilent. He is a mass spec expert. And while mass spec is routinely being used for primary structural characterization, I've invited him here, and let's hear from him before we go to the next question, how he can contribute for higher order structural characterization. So, Vadi.
Thanks, Navratna, for invitation and giving an opportunity to participate in this panel discussion. So, see, obviously, higher order structures, typically we think about AUC, DLS, and all these different things, NMR, all those things are very critical, right? And when it comes to the mass spec, and as you probably heard from many, publications or events are talking, multi-attribute methods, monitoring and methods, there are both of two different things. So essentially, like in a higher order structure as well, some of the slides I have seen, the HDXMS. That's people probably consider, but probably not routinely done because of its own complexity, right? So the one of the other things you can do like aggregate analysis, the mass spec can be used. But right now people are talking about or thinking about publications on native mass spec. It's not very popular, but that's coming. A lot of publications and a lot of people are doing size exclusion with the mass spec. It can be done, and actually 2DLC mass spec can be also used along with the ion mobility mass spec, and it will go another dimension. So these things as 30 odd instruments or analytical tools can be maybe combined with one instrument, maybe multiple types of separation. It can be done. So mass spec always primary structure, secondary structure, probably like post translation modifications always used, and the PTMs can glycosylation or other PTMs can impact on the secondary or higher order structures, right? So the mass spec is going to give what information? It's a molecular level information, right? Absolute mass, that cannot be done by any other technology. So mass spec always plays important role, and also like it will play in coming days. Maybe may not be there in any regulatory filing today as of now in the mass spec for higher order structures, but in coming days I think every single molecule will go in the mass spec space. So yeah, so I'll be happy to talk more on that if anybody particularly interested in that.
Yes.
Thank you.
So, question
Hello.
from the back.
Yeah.
Okay.
Yeah. My name is Dr. Arun from Sipla. So my question is to Dr. Ashutosh. During a small peptide characterization, we give the NMR data, that's 1D and 2D data for comparison with RLD. But nowadays, agency is asking about the Mahalanobis distance. So what is exactly why they are asking now? Is there any value addition to the due to this Mahalanobis?
So essentially, even in the RLD batches, there can be minute differences, right? So generally, or even your drug product, if you are taking 3 batches, there can be minute difference in NMR because you are now comparing more than 100, 200 peaks. So in Mahalanobis distance, or first they do PCA, and then after that, using the PC component value, you can do that Mahalanobis distance. So when you are doing PCA, you are first showing that there is a, like, if I showed you eclipse, there are each dot represented one RLD or one ISDP. Now in those there is some variation, but those variations are not meaningful if your product is matching very well. Now from there, if they are not meaningful, your Mahalanobis distance the value is going to be less. If there is a variation between these two ellipses, then the Mahalanobis distance value will be more. And that shows that how closely you have matched in meaningful statistical manner how the two product, your RLD and your product. That's how they are compelling all the people to go for Mahalanobis distance calculation.
So is there any percent variation difference with criteria to show the similar sameness? Percent variation?
Percent
how
variation? No, I would say like when you do the PCA, if like it's less than 2-3%, you will get all the.
Yeah.
But essentially, if you look at, if you are doing detailed structural characterization by using so many techniques, even a small variation will be picked up. So the variation mostly come near excipients region if your product is really nice. And in calculation, the excipients region is excluded because that can vary. Otherwise, if the product is made fantastically, your DM will be coming very nicely, less than 3.
Thank you, thank you.
So
My last question is.
I will just add on to whatever he said. Mahalanobis distance is a measure for closeness between the two sets of data. Now, if it is again product dependent, the number 3 or 3.3 or 4 or 5 is again a product dependent value, which you will establish from reference product data. Now, coming back to his question, he was also asking about whether CD or any other technique can be quantitative. Yes, just like NMR, how you compare to spectra, you can also do a quantitation based on Mahalanobis distance. Again, PCI assessment in this case will also be dependent on what frequencies you choose to demonstrate your product comparability against something else. And that is how you can make all these qualitative tools more quantitative in nature. Now, whether we want to do it or not, it is all our choice. But that's the way. And probably just like in NMR, agencies have started asking questions. Can you show demonstrate DM which is comparable? I think one day they will be coming for all these other quality techniques.
True. And my last question.
See, this is, as Dr. Ashutosh as well as Navratna has explained, this is how to understand how closely you are to the innovator product, the DM values. We are close in numbers, you are very close to this. However, there are some challenges with respect to small molecules, like where there are some terminal amino acids like histidine, etc., where small changes in the pH, it will cause a little bit chemical shift change. So that is where definitely will not fall within the 3.3, whatever is stated by the agency. However, it is up to us, see, to take it like the small pH changes which is allowed in the product pH range.
Yes.
But if you take that pH range, you will not going to pass the monobias distance.
Yes, it's true.
And you adjust the pH back to the RLD range and then study that it doesn't have any biological impact or impact on the bioassay for the set pH range, and then to do the NMR, adjust the pH and then show the similarity. So that is one way to represent.
Yeah. So last question, excuse me, please, to the Dr. Pradeep. As you told, that ST should be less than 600 or max to 700. So, but if there is a high content of excipients like phenol and some dextrose, so how to get rid of that then?
Yeah, that's a good question. We have faced these issues. The industry standard practice is 700, but we internally target less than 600 to get a better profile. Yes, there are going to be disturbances. There are going to be disturbances not only due to excipients, but even salt interferes.
Yes.
So even NaCl interferes at the wavelength of 180 to 190 around. So there are ways to remove. Buffer exchange is one way, but then that generates the question of before and after buffer exchange structurally you have to prove was it really a structural change or not. There are some columns which are specifically designed to remove some excipients like detergents or sugars as well. So there are methodologies, but then these methodologies come with a complexity of additional step of proving the sample preparation validation to show that it does not structurally impact whatever you have done. But the basis of all of this will be, it is a comparative study. We have to remember that. So whatever you are studying would be, you would be doing it with the innovator molecule as well. So if post that it is giving you the same structural information, because it is qualitative in nature, we can still justify to the agencies.
We have
Thank you.
We are only running late. 2 to 3 questions only, please. The speakers will be here, so you can ask later as well. So.
University from academics. The question is to you, as you have shown that the CD can be used for the stress degradation study also. So as rightly said by Dr. Ravi, that regulatory authority is now asking about the differences after stressing the samples, what we are getting when we are talking about the evaluation of biosimilar products. So how we have to conclude the results? For an example, at the without stressing, if the RLD product is matching with her or biosimilar product is matching with RLD, but when we do the stressing and then in the SECMAL or in the CD, if we get the difference in the pattern generated in the CD spectra of RLD as well as the biosimilar product, then how, on what basis we have to do the conclusion?
Okay. This is a very big topic for biosimilar manufacturers always. See, it is biosimilarity. It is not sameness. So it is not going to be same in all aspects or all attributes of whatever we analyze. There will be some residual differences. And how we justify these residual differences is what the agency is interested in. So these residual differences can be justified with orthogonal testing by understanding its impact on efficacy, binding kinetics, all of these aspects. So when you study all of this together, you can build a case of those residual differences not impacting. And we have to remember that the biosimilarity also clubs the clinical in equation. Yes. Right now, we are only speaking about analytical similarity here. So any such residual differences which cannot be justified using analytical tools, then we take help of the clinicals, right from the phase 1 till phase 3.
Now, my question is related to the analytical similarity not observed after stressing the sample.
Exactly. So which means there were some residual impurities which was not available prima facie, but on stressing it is. So that is not a desirable thing to have in the first case. And that is why when you do a testing right from the product development, you can eliminate this within the scope of your process. So that is one aspect of it. Otherwise, you do a lot of studies to justify. There are structure function studies that you can do to justify these residual differences as well.
Okay.
Usually long-term studies, when they are looking good, For that,
Ah.
you can always have a way to justify.
Just to add on, like, I mean, that's a really interesting question because that's what FDA is asking nowadays. So that's a real pain point for us. So only thing is, there are a lot of multiple ways to justify that. How harsh you will going to, degrade. That's a key point. So because as much as I expose to heat, as much as I expose to the light, these are the two critical conditions. Even precisely FDA is asking in the case of peptides. So you will going to have a really differentiated characters. If you are lucky, you will have a same pictorial representation. So I'm happy. If there is a differential, then as Dr. Pradeep is rightly stating, what is the differential? Whether in the case of CD, how much I'm varying. So there are multiple factors. So as Dama is rightly stating, adjust the pH. So that's a key. Otherwise, we will end up with a differential one. And in case of aggregates, if it is differential one, eventually when leading with a very different aggregate, then I think it's really difficult to go with. You have to answer with the in vitro kind of studies and moving into in vivo. So there are multiple related tests should be done in terms of safety, efficacy, and immunological perspective.
Just to add, these are all stress. So we are actually forcefully degrading the molecule. You have to remember that the rate of change of impurity formation is not going to be same from one sample to another sample, even within the lots of the innovator or you. So that is why even this has to be done in multiple lots to understand the differences there. So there may be one lot which may give rise to just 2% of increase, but there may be another which give rise and it goes to 3 to 4%. So even those variabilities should be accounted to when you justify these kinds of differences.
One last question, please, and
Thank
then
you.
we close the session.
Yeah.
Thank you.
Hi. Hi, sorry. My name is Himanshu. I'm from IIT Delhi. I want to ask a question to Dr. Rao, and even Dr. Bhat can answer that question. You said AUC was primarily used as a post-market tool, but now it's increasingly being used in product development, and even mass spec is being increasingly used to probe things like aggregation and stuff like that. So what is the driving force to actually gain that kind of information right in the product process development stage? What is promoting you to how much information do you need, and why are we using such sophisticated techniques right in the process development? What is driving the industry to do that?
So one is this is all a regulatory requirement to ensure the biological safety and efficacy of the product. So in earlier days, just people used to do as a sameness study at the end of the product. So people used to go with a lot of the chromatography based purity techniques, and then all these similarity aspects used to test at the end of the product and just before the regulatory submission. Where with the new requirements of the stress studies, etc., which arises basically small differences because of the process, the way you lyophilize the product, at what concentration you lyophilize the product, even it can create certain small sort of amorphous aggregates or fibrillation. These are all can be seen at the end, and then as a result of it, again you have to go to the drawing board, and then so your timelines will differ. It's most expensive job to go back and then understand these changes. That is where now it has become a product development tool. If you understand, do the XY matrix, and then what are the critical quality attributes can be addressed from the from the first step of the process, what are all it's going to impact, and then see to take it to the end. That is where it helps to navigate the right product development path.
Yeah, Navratan. One last question from my side. Yeah, Navratan, here.
Okay, only one. Please.
Yeah. Here I'm already asking a question, Navratan. This is Sridhar from Ravishankara.
Okay.
Yeah, this question is for all the NMR experts and regulatory experts on the DICE. So where the expectation on this PCA, principal component analysis. So we, as a CRO, we facilitated one study for one of the customers, where after doing this study, the technical team or the technical NMR experts, what they stated is the number of batches that you have given are not enough to give the comparable data. So is there any batch size that is determined, or can you just throw?
So I think minimum of 3 of each batch you need. More is better.
Okay.
right? So then even if little bit variation that is coming can be captured. So I would say minimum 3 batch is needed for each side.
Yeah.
If you are giving more, your data accuracy will increase. Because eventually you want to separate between components, PC1 versus PC2, PC1 versus PC2. You should not go beyond that. So I would say 3 is the borderline case. More is the better.
Yeah.
You may need more, probably 6.
Yeah, thank you. But in addition to that, wherever I'm doing, like the expectation, regulatory expectation, especially for the peptides, the expectation is to have the batches to early stage, mid stage, and late stage. There I can generate as a manufacturer, they can provide the samples, but same kind of data or the batches I will not be getting for the RLD. So in that case, how to, because definitely it will not match. If I have to compare these different life cycle batch versus the RLD, where I will be getting one lot or maybe the closely related lots. So how do we justify that kind of data?
So I can answer this from biologics perspective, biotherapeutic perspective, protein. Right?
Yeah. Yeah, this is a common problem actually. See, basically once you understand basically the methyl region and then the amide region, as Dr. Ashutosh mentioned, these are the two important areas for the basically to peptide the hierarchy structure, and then leaving all the excipient range and other the small components. So once you generate the NMR data, it is very, very important to understand the spectral overlays, which region it is not matching. So as I explained in the previous question, if there are small differences which are arising from the pH, etc., irrespective of what number of RLD lots you analyze, it is not going to match. Probably you'll have to look at it in a different dimension, how to take it that back. And certain small changes which arises, which are not due to the process components, and then that is where the number of lots probably will help. So in the recent requirement from the regulatory is like from each sample you measure only one measurement, not the multiple measurements, to have the better analytical variability. That is where sometimes 3, sometimes we may have to go to more as well. So there is no precise number to say that 3 will work or 6 will work. So some of the papers even agency use like 7 lots, 9 lots. So it is the generic industry where you keep on generating this data. At the end, from the product demonstration, you use all the lots data cumulatively to demonstrate it. That's where as a post-mortem, it is important to start accumulating the RLD data since beginning of the product development. That is where you get more and more number of RLD lots.
Yeah.
Just to add on, this is a critical point. You can justify by 2 ways. One is you can use your developmental batches data. So what we do is 3 exhibit batches versus 3 RLD is a classical way of having a comparison, but at the same time, it's the same question. We also got it. Okay. So we have to strengthen our argument with the developmental data, what I am having. So that would be about 10 batches data, right? So along with that, you can also, do a kind of statistical analysis for each test. That's the key. So that you can state that, okay, my 3 would be okay. With this 3, I'm stating that my product is having a good statistical, there is no significant difference in the statistical evaluation. Probably FDA might accept on this part.
Thank you.
I request you to have more discussion.
Yeah, definitely. So I'll bother Pradeep more
Has been
later.
standing for a while, so please. That's last.
Thank you. So I'm Nitya here from Sinjin. So before I post my question, I would like to give 3 answers. One to her is that Nano DSF, yes, we do use Nano DSF as a part of, the list of methods that for the maps. Number 2, coming to the question to Supriya, that do you use the orthogonal methods? Why is the requirement at which stage? So we use not only orthogonal methods, we also use complementary methods. Complementary techniques is also an expectation. For an example, H&Ws, the high molecular weight species, are expected or quantified by SEC, whereas the fragmentations or the low molecular weight species are taken from the CE methods. Okay. And coming to the question someone asked here about the AUC, why the advanced technique is used in the early stage of the process development. The reason is that it is a very quick and easy, as he described. It doesn't require like ACC, mobile phase preparations, column selections. It doesn't need so many optimizations to fix the method. That's the reason we want to go ahead with advanced methods. Secondly, in case at the end of the DS, in case if we find any anomaly, and to track the changes, where it is coming from, at which stage this aggregate started, or what is that? And that is one of the reason where the regulatory or the expectation is to start using the advanced techniques in the early stage itself, or in the upstream, or in the process development itself, not at the end stage. Okay, now I'm posting my question quickly. When we have a multimeric form as a product of interest, tetrameric form or hexameric form product of interest, so why do we reduce them, denature them, or make it as a monomeric form? Then we characterize. Are we need to characterize this in the monomeric form, or in the hexameric form, or in the multimeric form itself? Should we characterize the molecule for the structure changes? Both.
I can address it if you want. I mean, allow me. Yeah. See, the importance is what is the active state of the molecule. See, if my monomer is active in terms of its biological action, I have to demonstrate that even at the individual monomeric level, am I similar to the different product or not? Now, in the formulated drug product, am I comparable? So, for that, you have to establish that the multimeric state is also comparable. It is a typical example of insulin. Insulin monomeric state is active, but in the vial, it is actually hexameric, which is what you want because the stability of your product is actually defined by the hexameric state. So you require both. Same is liraglutide. Liraglutide function is only monomer. Semaglutide function is only monomer, but you want to keep it as stabilized in the stabilized state as it is. So both are required actually. That's the.
I appreciate that, Navratan. But my question is, if the product of interest itself is the monomeric form, multimeric form,
See, there are two parts. All the technique won't, characterize in its native form. So that's where there are 30 different techniques you need to look into. So, for example, as he rightly said, insulin as well as liraglutide, as a classical example, what he has shown is, it's there in the heptameric form. So we have to prove by AUC that it's not in other form, right? That's my nature. I'm not changing anything. But at the same time, I'm putting in the chromatography. Obviously, it will, break down into monomers. So I cannot, measure that. So So what they want from the FDA is how RLD behaves exactly. My product should behave. So that you need to prove. So in multiple situation, you are proving with multimer product as in situ multimer, and at the same time, in other techniques, we have to go with other. It's like differential factors.
So Navratna, if you
just
permit, can I help on that answer?
Hello, sir.
Okay. So it's the way I look at it is this: you have say 5 foot, you have 5 footballs, right? Among those 5 footballs, you have 4 golf balls, and that's a whole bunch of it, and it's in a basket. Now 4 golf balls are among those 4 golf balls. There are 3 red and 1 blue. Now you have your biosimilar product, other product. In that one, if the 4th ball, red golf ball is also blue, instead of one being the other color, when you take a photograph of it in the basket, you'll not be able to see it. So that's how it is. When you have a molecule which is multimeric in its state, in the technique, when you're looking at it, you don't know if there is a difference at the monomer level. And if there is a difference at the monomer level in that higher order or whatever bigger structure you're looking at, you'll not be able to see that difference. So it's about resolving the difference is the problem when you're looking at it in a multimeric state. So that's why you have to do at the monomer level as well.
Got it. In terms of biosimilar, yes, what you say is correct.